All right, I think we're about to get started just to keep respect of everyone's time today. Again, thank you for everyone to joining us our talk on what is your cell type MapMyCells. I'm Laurel Filer. I'll be your moderator here today, and we're joined by some amazing scientists to walk you guys through about MapMyCells. Slide. Next slide, Rachel. Oh, awesome. Just a reminder, this webinar is being recorded. And this is actually the 5th webinar in our series. So if you've missed any of the previous ones or want to go back, they're all on our YouTube channel. Especially since today we're going to be talking about taxonomies. And if you wanted some more foundational knowledge, such as what is a taxonomy or cell types, I really recommend our more earlier ones in the series. We're happy to take questions, especially if you're the people listening live. This is your chance to get some really great live questions in. Please post them on either the YouTube channel during or after. We'll have a separate Q&A, but we can also answer the questions during the talk. If you're on YouTube, put them on the YouTube channel. If you're watching a recording of this, please post them on our community forum, which is found in the YouTube description. And with that, Rachel is going to kind of explain just the references that are available, reference taxonomies that are available in MapMyCells. Scott and CK is going to kind of talk about MapMyCells. And then we'll have a Q&A session. But again, if you have any questions that come up during any time of the presentation, feel free to drop them in the chat. And with that, I will let Rachel take it.
All right, thank you, Lauren. Hi, everyone. I'm Rachel, and I'm going to be going over a few of the reference taxonomies that are available in MapMyCells, and also just what a reference taxonomy is. So if you've attended some of our previous webinars, you know that at the Allen Institute, there have been multiple very large taxonomies generated over the past few years. And especially the whole mouse brain one is our really most recent mouse work. And we've gone into some detail in our previous webinars about how those taxonomies were made, but today I'm just going to describe what types of cells exactly are available in that taxonomy. And a reference taxonomy is something that you can reference for your own work. So let's say you're doing your own transcriptomic work and you have some cells that you're studying and you're not sure what cell type they fit in or what characteristics they have. You can map your data using MapMyCells onto our reference taxonomy, which is a very robust and high quality controlled taxonomy to get a better idea and a better understanding of your own data. And so as I just mentioned, there are 2 taxonomies available right now in MapMyCells for this purpose. One of them is the whole mouse brain, and the second taxonomy is an Alzheimer's, human Alzheimer's specific taxonomy called CAD. And so first, I'll be briefly going over what the whole mouse brain taxonomy has. And this was published last year. Yao et al. 2023 is the reference, and this was also part of a very large paper package. But the idea of this taxonomy was to profile cells across the entire brain. So previous taxonomies at the Institute focused usually on one cortical area or another area of the brain, and the goal was to profile the entire brain. And so this was divided up into our main brain region, so the forebrain, midbrain, and hindbrain. And then within those areas, you can see we have more divisions. And here in this image, this is color-coded to match the Allen Brain Reference Atlas, which was made a number of years ago. And here you can see that it's the same color scheme is used. So if you've been used to flipping through our reference atlases in the past, you will see that it's the same coloring here just to try to keep everything simple and straightforward. And so here we have all the different areas that were profiled, and there were multiple ways that the brains were profiled. So one was by doing micro dissections on all these different areas and running single cell RNA sequencing. And this was done on millions of cells from about 300 different brains. And then once all of those cells were collected and analyzed through RNA sequencing, then those clusters, they were defined into discrete clusters to see where they fit genetically and how they're related to each other and which cells are not related to each other. Then the next step, there was also spatial transcriptomics experiments done for this taxonomy, which involved using MERFISH or multiplex error robust fluorescent in situ hybridization. And that's a part that we'll get to a little bit more in the next webinar next month in ABC Alice. I'll be talking more about the MERFISH and the spatial transcriptomics, but that was just another way to look at the spatial distribution of these cells. And then all of this data was then aggregated together to create this taxonomy and to annotate it based on all of the cells collected in the whole mouse brain. So what did this end up looking like? This is the full taxonomy. This is a massive figure from the paper. This is a dendrogram. And this dendrogram here is representing all of the distinct cell clusters that were defined and identified in this taxonomy. And you can see their hierarchical relationships. And so at the beginning of roughly, I think between 7 to 8 million cells were originally collected for this experiment, but only, about 4 million made it through after very strict quality control. So these are very high quality cells, very high quality data, and this was the end result. And the way that this dendrogram is broken up is it's broken up by hierarchy. So at the very top here, we have 7 different nodes, and these are called neighborhoods. And then here, I'm going to go talk about one of these neighborhoods more. So one of the neighborhoods here on the far right is looking at the pallium. And this has, within the neighborhood, then we have classes of cells, then subclasses, then supertypes, and then clusters. So those are the hierarchies in this particular taxonomy. And so if we just highlight just this one neighborhood here, here's a UMap showing all the cells that were collected and analyzed for this particular neighborhood. So this is one of our neighborhoods, specifically looking at the pallium. And as I mentioned in the previous slide, we also had MERFISH spatial transcriptomics experiments done at the same time or during these experiments. So here is where those cells are actually represented in the brain. So on the left here, we have the UMap showing all of them. And then on the right, we have our spatial distribution of these cells. And so after we look at the neighborhood, the next step is to look at the class. The class is the next level on this taxonomy. So if we go back to the major dendrogram, here we have our class highlighted here, and there were 34 classes that were defined within the greater taxonomy. And here again is a UMap color coding all 34 of those different classes of cells. And then below that, then the cells were split after the class into subclasses. So when we go down to the subclass level, which is listed here, there were 338 subclasses. And then they were split further into supertypes and then clusters. And the dendrogram does not show the 1,000 supertypes and the 5,000 clusters because there just is not enough space to see all that. But this is just a walkthrough, and this is a figure in the paper, and this really just shows you the amount of detail and the amount of data that is available in this taxonomy. And so the second taxonomy that I'm going to talk about is the CAD taxonomy, and this is specifically looking at postmortem samples from patients with Alzheimer's. And so the Alzheimer's data is a massive collaboration between many people at the Allen Institute, many people at local hospitals and other areas, and neurosurgeons to collect this tissue and to handle this tissue and to process it. And one of the lead scientists on this project, Kyle, he actually spoke a little bit about this project in our previous webinar on January 31st, titled "What is a Taxonomy?" So if you want to hear a little bit more about this project, I would highly recommend going back and watching that previous webinar. And so just to give an idea of what is all here in our CAD taxonomy and in our cohort, there were 84 donors spanning the entire spectrum of disease severity, and a number of different measurements were done on this tissue. There was quantitative neuropathology, there was single nucleus omics, and also spatial transcriptomics. And a taxonomy was created from these samples as well. And here's what that taxonomy looks like. So this is specifically, it's not the whole brain, unlike our previous, slide from the whole mouse brain, which had many areas. This is just looking at one particular area. And we have our split between our neurons and our glia here. And what's really interesting about this particular taxonomy is that there are cell types that exist in the Alzheimer's samples that do not exist in the reference non-Alzheimer's samples. Right, so a new taxonomy was created and expanded upon to include those new cell types. And those are seen down here, particularly in the glial cells that have this CAD label down at the bottom, were new cell types that were identified in the Alzheimer's samples. And again, to learn more about how to mathematically determine determine what is a new cell type versus, what is bad data or something, I think it's helpful to look at our previous webinar where Kyle really goes more in depth into determining when you truly have a new cell type in a new sample. And so these are the taxonomies that are available right now in MapMyCells that you can then upload and map your own data to. And again, here's an arrow pointing to those special CAD cell types that were not present in the control non-Alzheimer's samples. And again, how are these taxonomies made? What's the math that goes into making them? This was discussed in our previous webinar. And if you want to view and interact with these taxonomies, both of these are available in the Allen Brain Cell Atlas or the ABC Atlas, where you can go and filter by specific genes, by different cell types, by different areas of the brain. And we'll be going much more in depth on how to use ABC Atlas in our next webinar on May 22nd. So both the whole mouse brain and the Alzheimer's data set are both right now in ABC Atlas. And both of these data sets have single cell or single nucleus data and spatial transcriptomics data. So you can look at them side by side and really dig through all of that data yourself. And coming soon, currently right now, we also have the whole human brain, which is single nucleus data from the Saletti et al. Paper that is currently in the ABC Atlas and will soon also be available as a reference taxonomy in MapMyCells as well. All right, and so that's just a brief overview of the taxonomies that are currently in MapMyCells or will be in MapMyCells. And next, I'll pass it along to Scott, who will discuss in much more detail how to use MapMyCells.
Thank you, Rachel. Let me get everything configured. Am I good to go? Okay. Hi, my name is Scott Daniel. I'm a data scientist here at the Allen Institute, and I am here to talk to you about what MapMyCells is. In the most cartoonish way imaginable, MapMyCells is a tool that we put out in autumn of 2023. My colleague CK Lee is here with me to answer any questions that I can't answer because either they involve a part of the design process that I wasn't involved in or because they involve the R language. Full disclosure, I am first, foremost, and always a Python user. Sorry about that. The outline of this part of the talk is going to go in 3 sections. First, I'll just talk about, in the broadest way imaginable, what is MapMyCells? What do we think it should be used for? What might you think it should be used for? Then we'll talk about how do you use MapMyCells. I'll show you the very slick web interface that our UX team put together, and then also where the code running on the back end lives in case you want to clone it and run it yourself on your own local compute for whatever reason. And then finally, we'll talk about what MapMyCells is doing, what's going on under the hood, what algorithms or algorithm does MapMyCells use to assign cell types to the data it is given. So first, what is MapMyCells? Why should I use it? We designed MapMyCells to answer the question broadly. I, as a researcher, have cell by gene data, and I want to know what cell types the cells belong to, according to the taxonomies supported by the Allen Institute. Some more detailed use cases that I could just think of with 15 minutes of brainstorming or something like, I have transcriptomic data or spatial transcriptomic data. I want to know what cell type each cell belongs to. And then I want to look at how those cells are spatially distributed and see if there's spatial segregation in the cell types in the brain. This is an actual use case. This is what was one of the steps that was done in the Yao et al. Papers. They ran spatial transcriptomic analyses, took the data from our MERFISH rigs, mapped the cells to cell types with the MapMyCells algorithm, and then plotted the cells as you see on the right here. These are cells plotted in MERFISH sections, colored by their cell types, and their spatial structure there. That's pretty neat. Another possible use case, maybe you have data that already has cell types assigned to it, either from another taxonomy, another method, and you want to map it with MapMyCells and compare the two cell type assignments, comparing taxonomies. Is there alignment between the cell types? Does that say something fundamental about the cell types in the brain? Maybe you're very ambitious and you want to compare across species. Take human data, map it to a mouse taxonomy, map it to a human taxonomy, and compare the two mappings and see if there's a conserved cell type across species. You could do that. Or maybe you have developmental or disease data or data, some other data in which the stage of the cell is changing with some axis that looks like time. You could map each of your data sets at each level of progression with MapMyCells and look at how the distribution of cell types evolves with your time-like axis. Again, these are just use cases that I came up with just sitting and thinking all by myself. Anything where you have cell by gene data and you want to know what cell types do the cell belongs to, that's what MapMyCells is built for. So great, you've decided you want to use MapMyCells. Where do you go? These URLs will take you to the web tool. The first URL lands you in the broader MapMyCells ecosystem, where we have lots of documentation about what MapMyCells is doing. We have example notebooks showing you how to download data and convert it into the format MapMyCells expects. Like, data comes in all different shapes and sizes, and choices had to be made about what MapMyCells would accept. And then this final URL is the actual web tool, which will process MapMyCells, which I'll show you in just a moment after we talk a little bit more about data. Okay, so you've decided you want to use MapMyCells. How do you do that? So MapMyCells has a few requirements on the data that you give it. The most qualitative requirement is that it has to look like cell by gene data. If you can reasonably describe your data as a matrix in which every row is a cell and every column is a gene, congratulations, MapMyCells can process that. MapMyCells will try to assign a cell type to every row in that matrix. Notably, it will try to assign a single cell type to every row in that matrix. I'm saying that for the bulk sequencers out there. There's some debate about whether bulk sequencing data is appropriate for MapMyCells. So as long as you're comfortable with a single cell type assigned to each row in the matrix, go for it. And then each row is mapped independently. What the mapping that is done on row 1 has nothing to do with mapping that is done on row 2. So your data can be as heterogeneous as you want it to be, as long as it is a matrix where rows are cells and columns are genes. This means MapMyCells is definitely designed for single cell sequencing data or single nucleus sequencing data. It's definitely designed for spatial transcriptomics data. It might not be designed for bulk sequencing data, with all, given all the caveats I just talked about. At a technical level, the requirements on the data are that it be in an H5AD file. H5AD, for those who don't know, is a library built on top, or it's a format built on top of the HDF5 format that is meant to support AND data objects. AND data is short for annotated data. If you Google AND data, you'll go to a Python library that supports the format. What it is a format that was designed to help you carry around your data and your metadata in a single structure that is fairly easy to share. We have a tutorial here that talks about how to convert, say, a CSV file of cell by gene data into an H5AD file. And there are other tutorials in our documentation for converting other formats of files into H5AD files. For those who know about H5AD files, the other requirements imposed on it are the data has to live in the X element of the H5AD file. MapMyCells knows nothing about H5AD layers. The rows have to be cells and the columns have to be genes. I keep coming back to this point because apparently that's backwards from what is traditionally done in R. R likes to put genes in rows and cells and columns, so you'll have to transpose it to put it into MapMyCells. The data is expected to be in the form of raw gene counts per cell. If you give us log normalized data, MapMyCells will map it, but we don't vouch for the validity of the results the way we do for raw counts. So it can be processed, it expects raw counts. Your genes need to be identified according to the taxonomy that you're mapping to. So if you're mapping to a mouse taxonomy, your genes should be identified with mouse gene identifiers. If you're mapping to a human taxonomy, they should be identified with human gene identifiers. There are ways to go from mouse genes to equivalent human genes. I've linked a GitHub repo down there that some Allen Institute scientists have put together to help you perform that ortholog mapping process. MapMyCells is not smart enough to do that on its own. We leave that to the user if you want to map from one species to another. And then finally, I would just like to say, if you can give us ensemble IDs for your genes, that is preferred. We had to make a choice on how MapMyCells would refer to genes in a stable way in the backend, and we chose ensemble. If you give us NCBI gene IDs or gene symbols, MapMyCells will do its best to map those to ensemble IDs, but we just hard-coded in a lookup table with all of the mappings that we knew about, a guaranteed way to ensure that MapMyCells knows what gene you're talking about is to use the ensemble ID at the beginning. You can give us NCBI and genes or gene symbols. I just recommend ensemble IDs. And then finally, the file itself has to be less than 2 GB in size. This is a requirement placed on us by modern browser architecture. But it's just a requirement on the file size. You can use whatever amount of trickery with sparse matrices and compression that you want to fit as many cells and genes into that 2 gigs as you can. The file itself just has to be less than 2 GB. So you go to that link I've been broadcasting, and this is what you're going to see. This is our web interface. Before I get into how to use it, I just want to call out on the right column, there's that learn about available cell type references, algorithms, and output files. That's a link. And that'll take you to some very detailed documentation we put together about the taxonomies that are supported, the data sets backing those taxonomies, the algorithms that are supported, and when we recommend and for whom we recommend each algorithm. Once you know what taxonomy and algorithm you want, using MapMyCells is really as simple as these 4 steps. You drag and drop your file to the point where it says drag and drop. You pick the taxonomy you want, you pick the algorithm you want, and then you click the big friendly start button. Wait 10 to 15 minutes, and the big friendly start button becomes a bigger and friendlier download results button. And that will give you a zip file containing all of the output files that MapMyCells produced while processing your data. Before we talk about that, I just want to talk about what happens if something goes wrong. I don't think it will. The code's pretty robust at this point. We built a lot of guardrails over the last 5 months. But if something goes wrong, you're still going to get a download button, and that will give you a zip file that contains a text file with all the error messages that MapMyCells produced on its way to crashing. And maybe that will be enough information for you to debug what happened, fix your data, reformat your data, and try again. If it's not, in the mapping failed dialog box, you'll see this long alphanumeric run identifier. Make note of that. You can go to the Allen community forums, post a message in the MapMyCells topic, and if you include that run ID, we can go look at the back end, get a more detailed bug report than maybe you got, and help you figure out what needs to be done to kind of to recover your run, to get your data through MapMyCells. So that's what to happen. That's what you should do in the unlikely event that the thing just crashes. But it probably won't, and you'll get something like this. Like I said, you'll get a zip file that contains these files. It's all documented here in this link, but I'll talk you through what these files all are. So first, there's the validation log.txt file, which again, that's the log file I talked about in the event of a crash. Just contains all of the warnings and status messages that MapMyCells tried to write to the terminal while it was running. There will be then 2 files with a long identifier that say something about your data and a timestamp, and then .csv and .json. And these are the main event. These are the mapping results. The CSV file is kind of our flagship output product. It's just a flat table. Every row is a cell, every column is a cell type, and it just tells you what cell types your cells were assigned to. The JSON file contains that data along with some richer metadata. It contains the config parameters that were sent to MapMyCells. It contains a lookup table of the marker genes that were actually used in mapping your data. And it contains a bit more about the mapping results. So in our algorithm, there's this concept of like runners up, cell types that were almost assigned, but not quite for some reason. And those runners up will be listed in the JSON file, but not in the CSV file. We didn't want to blow up the size of the CSV file too much. And then finally, there's a summary metadata.json file that just contains number of cells mapped, number of genes mapped, genes used in the mapping. That's mostly for our bookkeeping, so we can see how much data is getting pushed through MapMyCells, but we give it to you anyway in case you want it. So let's talk about what's in that flagship product, that CSV file. Like I said, it's just a flat table. Rows are cells, columns are cell types. You can load it in Excel if you want to, which is what I have done for the purposes of visualization. The first few rows are just metadata. It tells you what detailed, first it gives you the path to the JSON file associated with the CSV file. So you can look up that richer metadata in the JSON file. Then it tells you what taxonomy you're mapping to. What are the levels in the taxonomy? These are the class, subclass, supertype, cluster that Rachel was talking about in the case of whole mouse. And then it tells you what algorithm was run, where the code lives and what version of the algorithm was run, just in case you run it and then 6 minutes later you come back and run it and maybe we updated something. And then you're off to the races. Then you have your data. There's this table. Every row is a cell. It's identified with the index column from the OBS data frame in your H5AD file. And then the columns represent the cell type assignments for each of those cells. There are machine readable assignments. These are accession IDs that have been assigned to the cell types. They're a way for us to stably and for all time refer to the cell types in a given taxonomy. And in the future, these will be a good way to interface MapMyCells results with something else in the brainmap.org brain knowledge platform ecosystem. But that's kind of a future use case, and maybe you don't want to deal with the machine readable cell types, which is why we also give you the human readable labels for the cell types. These are the slightly more scientifically meaningful names of the cell types to which each cell was mapped. I guess I forgot to point out that if you look at this table, you'll see that there are, in this case, 4 cell types for each cell, and that's because the cell was assigned to a cell type at each level in the taxonomy. There's a class the cell was assigned to, a subclass it was assigned to, a supertype it was assigned to, and a cluster that it was assigned to. So you get that for each level in your taxonomy. And then finally, there is a confidence metric, a number telling you how confident was MapMyCells in the mapping that it did. Depending on the algorithm you choose, this will represent different statistical quantities, but it's all on a 0 to 1 scale. 1 means MapMyCells was perfectly confident. 0 means MapMyCells threw up its hands and had no idea what it was doing. So that's a way for you to put a quality control cut. Which cell type mappings are you willing to believe for the purposes of your analysis? You can do that with this numerical quality metric that we provide you. Yeah, so that's everything that you'll get in your output CSV file. And like I said, all of that is also in the JSON file, along with some more detailed results for those who feel confident working with a JSON file. Just so you know, if you feel comfortable in Python and for whatever reason you don't want to go to the web tool, maybe the 2 GB file limit feels claustrophobic to you, we have made this code publicly available on GitHub. There's the repository, there's documentation on how to use it in the repository. And I also just want to call out that there's documentation for ingesting or preparing your own taxonomy to run with MapMyCells in the repository. Let's say you have a taxonomy your lab is working on, or maybe a taxonomy that a collaborator lab is working on, and you want to map to that taxonomy using MapMyCells. You can do that if you clone the code and run run some preparation scripts that we've tried to make as user-friendly as possible. So just that option is available for those of you who feel comfortable in Python and have access to compute resources of your own. Okay, so that is how you use MapMyCells, but what is it actually doing under the hood? So we right now support 3 algorithms in MapMyCells, and that's something that you choose from on the web tool UX. There's a deep generative algorithm, which uses a variational autoencoder to map the reference taxonomy data to your unlabeled data into a latent space, and then assign cell types based on proximity in that latent variable space. There is the hierarchical mapping algorithm, which uses a Pearson, which just correlates each cell with the average gene expression profile of each cell type, and then assigns the cells based on how correlated the cells are to the cell types. And it does so in a hierarchical way, first assigning a class, and then assigning a subclass that descends from that class, and then assigning a cluster that descends from that subclass. And then there's flat mapping, which is just hierarchical mapping, but it skips the hierarchical part. Instead of assigning class, then subclass, then cluster, it just goes straight to cluster and infers the parent levels of the taxonomy from the cluster that was assigned. I'm going to talk to you about what the hierarchical mapping algorithm is doing, both because that is the algorithm we have made available through GitHub, and also because it's the algorithm that I was a part of implementing, so I understand it better. Before we talk about what the algorithm does, I just kind of want to set the table what the algorithm knows. So the hierarchical mapping algorithm depends on 2 summaries of the taxonomy data. It depends on the average gene expression profile of each cell type in the taxonomy. And then it depends on a marker gene lookup table. Both of these things we have already given to MapMyCells. And if you were preparing your own taxonomy, you would generate those files as described in the GitHub repository. This marker gene lookup table is a little different than people often talk about marker genes. What it does is it maps a class, or it maps a cell type to the list of marker genes that are good at discriminating between that cell type's children. So, if I say class A, list of marker genes, it doesn't mean those marker genes are good at identifying class A. It means those marker genes are good at telling the difference between the subclasses that are descended from class A. So it's a slightly different way of talking about marker genes than sometimes I hear people talk about. Yeah, so once MapMyCells has those 2 summaries, in broad strokes, what it just does is it takes each cell, and it correlates the cells with the cell types using the marker gene in the space of the marker genes, and picks the cell type that has the best correlation. It's a little more complicated than that, which is why we're going to walk through a cartoon. Let's say we have this very simple taxonomy, and let's say we have a cell that we know is a cerebellar neuron, but we want to know what it is below that. We want to decide, is it a cerebellar glutamatergic neuron or a cerebellar GABAergic neuron? If it's glutamatergic, is it a granule cell or a DCO nucleus UBC cell? If it's a DCO UBC cell, is it cluster 1 or cluster 2? How does hierarchical mapping make those distinctions? And what it does is it walks through the tree with the following process. First, you look up the marker genes associated with cerebellar neuron. Recall those are marker genes that are picked because they are good at discriminating between the children of cerebellar neuron. Then it randomly selects 90% of those marker genes, randomly selects a subset, and it correlates the cell with the leaf nodes of the tree, so the very bottom of the tree, in that space of the 90%, the randomly selected 90% of marker genes. It will then assign the cell to the leaf node with the highest correlation. However, because we aren't actually assigning to the leaf node, we are just asking, are we a glutamatergic neuron or a GABAergic neuron? It treats that assignment as a vote for the relevant parent. It treats a vote for assignment to granule cell cluster 2 as a vote for cerebellar glutamatergic neuron. Why did I talk about votes? Because we're going to repeat this process 100 times. We're going to iterate 100 times, every time picking a different random 90% of marker genes, correlating with the leaf nodes, finding the most correlated leaf node, and then assigning a vote to the leaf node's parent. And so after we do this 100 times, we will look at the parent that has the most votes and say the cell must belong to that parent. So if we did this 100 times, and 81 of those times the cell was assigned to some child of cerebellar glutamatergic neuron, we would say, okay, the cell is a cerebellar glutamatergic neuron. Now we have to decide, is it a cerebellar granule cell or a dorsal cochlear nucleus UBC cell? And so we repeat the process, except now the relevant parent, or the grandparent, is a cerebellar glutamatergic neuron. We look up the marker genes associated with that. We randomly select 90% of them. We correlate with the leaf nodes only that are descended from cerebellar glutamatergic neuron. We're going to ignore the right-hand side of the tree because we've already ruled it out. And we're going to repeat that process 100 times. Now, when we find that the cell is most correlated with, say, DCO UBC cell cluster 1, we treat that as a vote for dorsal cochlear nucleus UBC, put a vote there, repeat 100 times, figure out whether granule cell or DCO UBC gets the most votes, assign to that, and then try to assign a cluster. Just walk down the tree until the cell has been assigned to every layer in our taxonomy. So that's what it does. How well does it do it? Asking that question is difficult because in order to say how accurately MapMyCells is working, you have to know what cell type your cell actually belongs to. And we are all here because knowing what cell type your cell belongs to is hard. And so to do the test, what we have done is we've taken a subset of data from the Yao et al. 2023 release, and we're going to treat those annotations as correct. Those annotations were done in a totally different way. They took the data, they clustered the data ab initio with a bunch of complicated linear algebra, and they learned the cell clusters from the data. That is, in fact, how the taxonomy, the whole mouse brain taxonomy, made it into MapMyCells. So we'll take random 800,000 cells from Yao et al., and then we'll run that through MapMyCells, and we'll ask the question, did MapMyCells reproduce the ab initio annotations in Yao et al. 2023? There's another test that we also want to run. As I said, MapMyCells has in it a lookup table of marker genes that we gave it. And that's all well and good, but what happens if you, the user, didn't measure all of the marker genes that we told MapMyCells to expect? How well is MapMyCells going to perform? And so in addition to the test I just described, we are going to take our test data and mangle it so that it only includes a random subset of marker genes and see how that affects the results of the mapping. And this is what we found. So horizontal axis is the number of marker genes in a test set. The total number of marker genes that MapMyCells expects is 6,558. Vertical axis is the average F1 score. It's just a robust way of talking about the accuracy of the mapping. 1 is perfect, 0 is always wrong. For each number of marker genes, we ran the test 10 times. So we randomly selected 10 different subsets of marker genes. The points are the median of the F1 score for those 10 iterations, and the bars extend from minimum to maximum. And so the big takeaways for me from this figure are, 1, if you have all the marker genes, MapMyCells does very well. It has almost perfect assignment at the class level, 97% accuracy at the subclass level, more than 90% at the supertype, and about just over 80% at the cluster level. But additionally, if you don't have all the marker genes, if you only had half the marker genes, you still get basically that result. Like maybe it's closer to 95% at the class level and 93% at the subclass, still better than 90% at supertype and just under 80% at the cluster. So you can get away with a lot, not all the marker genes that we have. And things only really get a little scary if you go down to 1,000 or even 500 of our 6,000 marker genes. However, I just want to point out that these marker genes were randomly selected. I did nothing clever. I just put them in a random number generator and shuffled. If you do something intelligent, which I suspect you will when you're designing your gene panel, you can do very well. And I verified this by taking the gene panel from our MERFISH data, so which is 500 genes, all of the markers because they were selected with foreknowledge of the Yao et al. Taxonomy that we knew we wanted to map to. And then took the Yao et al. Test data, those 800,000 random cells, downsampled them to only have the 500 marker genes from the MERFISH panel, and did the mapping. And again, we got basically the same results as if we'd had all the markers: 98% accuracy at class level, 97% at subclass, 90% at supertype, and 77% at cluster. So if you choose well-chosen genes for your gene panel, you can get very good results out of the MapMyCells mapping algorithm. And so finally, I just want to talk about the quality metrics that this algorithm implies. How does MapMyCells communicate to you its confidence in the mapping? And there are two metrics that come along with the hierarchical mapping algorithm. One, the average correlation metric, and that average correlation coefficient. And that is just, you have these 100 iterations of mapping. You pick those that chose the final cell type, and then you average their Pearson correlation coefficient. How correlated was the cell with the chosen cell type in the iterations where that chosen cell type was actually chosen? High correlation is good, low correlation is bad. Then there's what we call the bootstrapping probability, which is what fraction of those 100 iterations chose the final cell type. Was the final cell type chosen in an 81-19 vote, or was it chosen in a 51-49 vote, or a 40-30 vote? Those differences have an implication for how much you should or maybe want to trust the cell type mapping. In the CSV file for hierarchical mapping, you just get the bootstrapping probability, but in the JSON file, you do get both. And so you can choose where you want to make your cut. Yeah, so that's MapMyCells. How to use it, why to use it, what it does. I'm just going to leave these, throw these links up here for a moment. Again, these first links are the MapMyCells ecosystem on brainmap.org. There's the community forum page, specifically the MapMyCells topic, where you can post any question about MapMyCells, and people in charge of keeping MapMyCells alive will see it, and we'll try to answer it to the best of our ability. And then there's the code on GitHub for those of you who feel ambitious and want to run it yourself on your own compute infrastructure. Thank you for your time. Happy mapping. And I will now take any questions.
Thank you, Scott, for that great presentation. As Scott said, happy to take questions both on YouTube or on Zoom. In the meantime, quizzes. Just launched a really quick, just if this webinar was helpful, this help us keep track of if these webinars is useful. You know, Scott's time is precious and we want to respect that. So if you don't mind filling that out, that would be great. But if not, feel free to post questions in the chat, either on Zoom or YouTube. Oh, we got a question from YouTube. Thank you so much for the presentation and working on a handy tool. We in the field really appreciate it. Would we have a recommended way to join two different annotations, example CAD plus custom?
I guess what do you mean plus by join? Like, yeah, can you elaborate on that? It's you map to CAD and then you map to a different taxonomy and then see how those two things compare? Is that what you're asking?
I will see what they say back on YouTube if Felipe wants to answer that. Give him a moment, but if not, we also do have another question. I assume this might be a shorter question. In the meantime, from the chat on Zoom, great presentation, thank you. Do you have a specific timeline to include the whole brain human reference from Soletti at all? Really looking forward to it.
It's specific in my mind that by the end of Q2, so that's late June. Yeah, by the end of June, we ought to have it in there, but a firm deadline has not been set.
All right, going back. Yeah, if the Felipe from YouTube wants to clarify what does he mean by joining, happy to answer that. But if not, no worries, you can always come back to our community forum to post that question.
Yeah, sorry, I just.
No, you're good.
Yeah.
Oh, got another question from Zoom. Is hierarchical mapping assign unknown cells at the final level? Oh, in hierarchical mapping, assigning unknown cells is the final level. Is there advantage to systematic hierarchical classification?
I think so. CK, you did the horse race between hierarchical and not hierarchical. So do you want to speak to that?
Yeah, for when you do mapping, let's say across a species or across platform, I think we recommend to the hierarchical mapping. And but if you have for example, our the whole mouse brain taxonomy is built on the 10x version 2 and 10x version 3. So if your data is from that same platform, you can just use it the flat mapping. So and both case it goes down to the cluster level, but I think the usually at the subclass level is I think everybody is comfortable with. Yeah.
All right. Join person has not one. Someone has their hand up, so I guess I'll allow them to talk on that.
Go for it.
Hello.
Hi, sorry. I'm gonna put my hands down because I found the Q&A
Oh,
panel.
no worries, no worries.
Thank you. Bye. See you later.
All right. Oh, here it is. Her question. Is there that's a lot. Is there a version of these slides or other doc that we can click on these links? I guess on the version, yeah, on the question on about the slides. Oops, opened. Is there version slides? We'll figure out on sharing them, but all the links I think Scott mentioned, a lot of that was on the community forum. Map said learn about. Unless Scott, is there any other forum on that?
Yeah, yeah, they all go to the brainmap.org. So if you were to go to brainmap.org and click through like to cell types to mapping, you'll get to all the documentation from MapMyCells, with the exception of the GitHub repository, which is just Allen Institute /cell_type_mapper. And yeah, but even brainmap.org eventually will link you to that repository because some of our documentation lives there. Yeah.
I will post it to everyone in the chat of that. And also, again, will be recorded on YouTube. It should be hopefully by YouTube the end of the day with some minor tweaking of just awkward silence in the beginning, but you can always pause there on YouTube and grab the links there as well. Next question. Great presentation. I see that the map result label is quite abbreviated. It's like ENTMV, /PA-COP-glute. For example, is there a translation spreadsheet for the class, subclass, supertype etiquette?
Yeah, in the taxonomy page, I think we should have a Google Doc for each the subclass, the class, subclass, supertype, cluster level. Everything is annotated, so you can look at it for the detailed information about the labels.
Yeah, I think the way, because that will all be, so all of this is for whole mouse brain, it's all based on the OUTL 2023 data, and so that ought to be accessible through the ABC Atlas whole mouse brain release. MapMyCells itself won't link to it, but if you go to brainmap.org and look for the whole mouse brain taxonomy 2023, you'll find links to how we release that data, and that will include links to the document that CK was just referencing.
Great, thank you. I think someone asked, will you align the cell types of CAD and SLUTty at all? Unless Scott or CK understands that question.
I don't, I think I do. And I think the answer is no. At least no one has told me that's yeah, I think we're, for the moment, we're going to treat CAD and SLUTty as independent taxonomies that the user can choose to map to or not. I think, yeah, for now.
They will be totally separate. Yeah.
Yeah.
All right, next question. Are there any modifications you would suggest for the Merfish data with smaller gene panels, example 150 genes, for example, using a smaller taxonomy? This would involve us using cell type mapper and not the web interface. Thanks.
Smaller taxonomy.
one way we are using it, when you if you know, for example, your data is from certain brain region or something, then after you run your map, run the MapMyCell, and then you can see the assigned cluster, and then if you look at the annotation table, then you will see this cluster supposed to belong to certain region. Then if it is out of your region, then probably you'd better filter those guys out, and based on the confidence value. Yeah, and then the next another way is you can select certain portion of taxonomy, and then you can do the MapMyCell. But I think I'm not sure whether the MapMyCell has that capacity yet, but I mean that's the best approach you can do. Yeah.
Great.
Yeah, so yeah, the web tool won't allow you to downsample the taxonomy, but there are ways in the cell type map in the actual code, I think. So yeah, I think what CK described would work.
Yeah, so subset the taxonomy from whole brain to only those region. Yeah.
Great. Let's see. Any other questions? I'm on YouTube. Give it a few more minutes. But if not, again, our community forum is really active. So if there's any other questions you have later at follow up. But again, if you don't have didn't get time for your question today, feel free to put in the community forum. I guess follow up, marker genes are weighted equally. Do you try using an algorithm that weighs certain marker genes higher than others?
That was earlier. Did we, CK?
Yeah, we didn't try weighted weighted markers. Yeah, we treat them equally, but at the hierarchy, different set of markers are selected. So I think it's been taken care of in that sense. Yeah.
All right. Any other questions? Not, I'm happy to give everyone 10 minutes back of their day. Oh, everybody says thank you. Last chance. If not, again, community forum. And also these slides will be available on YouTube. In the chat, we posted some of the links of the MapMyCells and annotation table if you want to access it. But again, that is also on BrainMap as well. With that, thank you again, Scott and CK and Rachel, for their time today. And thank you for everyone for joining us.
Thanks, everyone.
Okay, bye.